Welcome to my video lecture. Today we are going to discuss about enzyme immobilization. So what is enzyme immobilization? So literally we can say immobilization is nothing but arresting the mobility of an enzyme. In general, when you are not doing any immobilization, if you take a test tube where you had your free enzyme and your substrate, so both is freely moving in the container. So there is a possibility of collision where it will convert the substrate in the product. But this is a free enzyme reaction. So in this, what you are going to do, you are going to arrest the movement of an enzyme by attaching an enzyme to a solid support, either in this way or there is another methods are available. So basically you are arresting the movement in a fixed phase. But why we need to do that? Right? What is the purpose of that? Because as I mentioned, whenever the collision happens, then only the reaction might happen. That is, the productivity happens. But by reducing the mobility, basically you are trying to reduce that movement and reduce the number of collision happening between the enzyme and substrate. But why we need to do that? There are certain reasons why we need to do enzyme immobilization. And immobilization is not always needed. Only under certain conditions, when only then you can use the enzyme immobilization. What are that conditions? The first one is called enzyme reutilization. What is that? Why we need this? So enzyme reutilization is basically needed whenever the enzyme cost is too much. So basically we know that enzymes are not consumed in the reaction. So at the end of every reaction, the enzymes are remains there. Since removing that enzyme is a very tedious job, that's why normally we don't bother about the enzyme. We just separate the product and we move on. But what happens if the enzyme which you are using is costly? So if there is a way to reuse that enzyme, is always beneficiary. So like imagine you are attaching an enzyme to a solid support and then you add that into a substrate solution like this. So after the reaction is over, then you remove that immobilized enzyme from the solution like this. So what happens is you can reuse it again for the next cycle. So it is very much reduce the cost of the production. So that is the first need for the enzyme immobilization. What is the second need? The second condition is needed is the model for membrane bound enzyme. What is that meaning? There are certain enzymes which is only work better when it is adhered to a support. Example is the membrane bound enzymes, because it's naturally in vivo condition these enzymes are attached to something in order to have a better activity. So if you are separating those membrane bound enzyme and using in a free form, is always reducing the activity. And you cannot investigate the activity or any changes if you made in that is not truly reflecting if you use that in a free form. So always you want to use it in the attached form. So in that scenario, you can use enzyme immobilization. Right? It's actually used as it's a mimic the in vivo condition. The third one is a stability enhancement. It's a very least used, but also in some cases they are using to increase the stability. So what I mean that, in case if you are using an enzyme free form and if start degrading or melting say 50 degrees Celsius, but your process require to run the entire operation at 50 degrees Celsius or 55 degrees Celsius. In that scenario, you cannot use the free enzyme. The reason is immediately the half-life comes down at the 50 or 55 degree, very short half-life. So you can enhance the half-life of an enzyme by you put the enzyme in a productive layer, or you can entrap into the enzyme, so that you can very well use this. You can increase the half-life. So even at 55 degrees Celsius, the same enzyme work in a extended period of time. So this is not only applicable for the temperature, it's also some extent it is used for the different pH too. So these are the three important reasons why we need to do the immobilization. So basically you don't need to do all the time. Only when you need certain period you can use that. So this is what is enzyme immobilization. How we can perform enzyme immobilization? There are many methods are available. So they are actually broadly classified into two categories. The first category is called surface immobilization. What I mean by surface immobilization, your support material will be like this. So whereas your enzyme attached to this, the surface of your support material. So it is like that. It is actually added to the wherever this test container, you can add directly like this. So what are the methods that are available in a surface immobilization? The first method is available is called adsorption. So name suggests it just attached to the surface through very weak forces such as van der Waals forces, dispersion forces, hydrogen bondings. So it's a non-covalent interactions. So the major problem if you are doing an adsorption based is called desorption. Desorption means it's just these enzymes are leaked from the surface. So when the time progress. So how we can avoid the desorption? Desorption, one way to do that, you can use a glyceraldehyde based linkage, or you can do a cross linking between the enzyme like this. So even if this is this breakage is gone, because of this cross linking with the glyceraldehyde, these enzymes remains intact over the surface. So that is the one way you can reduce the desorption. You can only reduce, you cannot completely avoid that. So so that is a major problem here. And what is the material that can be used for surface immobilization using adsorption? Mainly you can use any inorganic material like alumina, silica, ceramics and glass. These are all the inorganic material that you can use as a support material. When it is come to an organic material, you can use a cellulose, starch, activated carbon, etc. There are so many other methods are there. So we are all this material can be used in a adsorption. So what is the next method that available? So in surface immobilization, the second method of surface immobilization is called covalent binding. That means your enzyme and the solid material is attached through a covalent bonding, right? So how this covalent bonding happens? There are function group present in both enzyme as well as the support material, like amino group, hydroxyl or carboxyl, sulfohydryl groups. So these groups is sharing the lone pair of electrons, and it forms a covalent bonding. In these type of covalent bonding also had a problem in covalent bonding. This kind of covalent bonding causes this problem. So what is this is called blocking of enzyme active site. So what I mean by that? See, if you are having an enzyme like this, imagine you have a SH group here as well as a SH group is here, sulfohydryl group. That's a functional group of an enzyme. Similarly, your support material having a functional group which we can form a linkage. So this is a functional group I am using R. So this can form a bond with the SH group. So what here will happen, what is the possibility here is, this R group can bind to this SH or it can bind to this HS. What happens if it is binds like this? That means the active site, because this is the active site. This is the active site. So this active site, now it's, you can see in this scenario, it's called blocked, right? Because your R group is attached to that. So even though the enzyme immobilization is success in this scenario, but that enzyme has become inactive because active site is not available for the substrate to bind. So how I can avoid this? Is a problem in covalent binding. How I can avoid this? So there is one way to avoid this is using competitive inhibitor, or we can say competitive inhibitor analogs. What is competitive inhibitor? The competitive inhibitor normally binds to the substrate of an enzyme, and whenever the substrate concentration is increased beyond a point, that got released. That is, this inhibition is reversible by increasing the substrate concentration. It is not like other inhibitor, so we can always remove this. How it help in avoiding this problem? For example, if you are having a same example, like you have an enzyme, you have a SH group in both scenario, both side, and before you go for immobilization, first what happen is you add a inhibitor, a competitive inhibitor. So you can say this is a competitive inhibitor. So what will happen? So this is a enzyme inhibitor complex. Now the active site is actually blocked or occupied by the competitive inhibitor. Now this is attached to the surface. Only possible way to attach is like this. Only this functional group is actually enabling the immobilization. This is the one trick that we can always do to avoid the problem of blocking of enzyme site. In most of the scenario, the functional group of a support is always activated, need to be activated many of the time with the help of many materials. So here the example is cyanogen bromide or glyceraldehyde. There are so many other strategies that we can activate the functional group. Because why we need to do this? Only when there is a need for immobilization, there is this functional group is become active, right? Till that it will not allow to do that. So it will somewhat protect that functional group from unwanted binding of any chemical with the support molecule. So this is one way to protect your functional group. So this is the two methods that we used in the surface immobilization. One is adsorption, another one is covalent binding. So this is one way. The second broadly classified method is called entrapment method. So what is the name suggest? The entrapment method is nothing but it is binds to the enzymes or binds or trapped into a closed enclosure. So here this is the thing. So here your enzymes like this. So physical enclosure in a small space is what we call entrapment method. Again, there is a different type of entrapment method. One is called matrix or gel entrapment method. So what is the example for that? Is what are the material that we can use is called a polymeric material like alginate, polyacrylamide, collagen. So alginate has a wide application or widely used method. The reason is it's very simple to perform. It's not very costly. It's a very cost effective method. How this calcium alginate perform in the lab conditions is easy. So the sodium alginate is a starting material, and if you look at that alginate has a peculiar character. The solubility of the alginate depends on the associated salt, that is solubility in water. So whenever alginate is associated with sodium, it is soluble in water. But if this alginate is associated with a calcium, like a calcium alginate, this becomes insoluble in water. So this is a property that we are going to exploit normally in the lab session. So how they perform the enzyme immobilization using this method? They take the prescribed amount of an enzyme in a solution form, then they mix that with the sodium alginate. So it has become a mixture, right? Normally sodium alginate is you can easily dissolve by heating the water. Once it's become cooled, then you can add enzyme, so it has become mixture. Then you take that in a pipette, it may be a micropipette or any normal pipette, and you add drop by drop this mixture into a calcium chloride. So what happens is, whenever you are using sodium alginate, if I mix that with the calcium chloride, so the reaction resulted with calcium alginate plus sodium chloride. So this change of salt immediately changes the property of an alginate. So it is immediately become a insoluble. So whenever it touch, the reaction is very quick and it form a beads. So the enzymes which is in the mixture is become trapped inside the beads. So it's like that. So you have a beads. So these are the enzymes which is trapped inside. So this is how normally we perform a calcium alginate based enzyme immobilization. It's generally called calcium alginate gel entrapment method. So what is the other method for entrapment that we call membrane entrapment method? So what is this? You need to take two membrane and your enzyme are in between the two membranes. These membranes are semi-permeable in nature. Right? So this is another method. So we can control what are the substrate to penetrate through by changing the porosity of the membrane. It is very easily to control in membrane entrapment in compared to the matrix gel entrapment method. We can control even in gel entrapment method by changing the percentage of calcium alginate. But where the gels are forming random pores, the size is not very regular. But in membrane entrapment method, you can choose a membrane which is having the regular pore size, so that you can compare how what are the things which is need to be penetrated through the membrane to access the enzyme. Right? So what are the different type of membrane that we can use is nylon, cellulose, polysulfonate, polyacrylate. There are so many other membranes. This is a common one which I mentioned here, but other things are available. The enzyme immobilization method is not always useful. There are so many other disadvantages we have. So what are the disadvantages for the enzyme immobilization methods? The first one is the enzyme leakage. So normally we do perform the experiment with a defined concentration of enzyme so that we can avoid the factor that influence the productivity. So, for example, if I am deciding to use 50 mg per ml of an enzyme for my reaction. So you are calculating that, and based on that you are preparing an enzyme immobilized enzyme. You think that it is useful and you are reusing it the next time, reusing it another time. So every time you assuming that the 50 mg per ml concentration is used in every reaction. But what actually happens, these enzymes are subject to leak. Leak means it's go away from the support. So initially your reaction container might have, the first reaction might have 50 mg per ml, but the second reaction definitely not having 50 mg per reaction, it might reduce to less than that. Normally 10 to 15% there is a loss of enzyme. So this is a major problem. So we are unable to control what is the amount of enzyme that we are using. So every time the varying concentration definitely affects the productivity. So that is become a huge factor that influencing that. So we are unable to predict. It's a problem. And which of the method is most predominant for the enzyme leakage, especially the adsorption method of a surface immobilization. So this is a major problem because there is no very non-covalent interaction is easily subject to detach. Then for extend your gel entrapment method also is easily get detached based on the porosity of the molecule, support molecule. So this is the first one. So first factor that affects the enzyme immobilization. The second, the most important method problem is diffusional limitation. We also call this as a mass transfer limitation. What is this mass transfer limitation? So very simple. In case of gel entrapment method, your enzyme is present inside the matrix or gel like this. If this enzyme want to convert the substrate into a product, there should be a interaction between substrate and enzyme. Normally the substrate molecule is present outside this in a reaction chamber. So if this substrate want to access to the enzyme, it has to penetrate this material, that is your matrix, in order to go into that. Whenever such phenomena happens, not all the substrate available in the bulk liquid can penetrate and access all the enzyme present in the beads. So this is causes the severe limitation in the movement of the substrate. Here the mass is nothing but the substrate. This is what we call a mass transfer limitation with respect to the substrate in order to get the access to the enzymes. It is a very serious problem because normally we predict any productivity based on the available substrate concentration and available enzyme concentration. Here the enzyme concentration may be present like that, but your substrate concentration severely reduced when it is penetrated in the like this. The substrate concentration is reduced like this. So this is the radius of So here the x-axis is radius, your y-axis is substrate concentration. At the radius progress, your substrate concentration is keep reducing from the periphery of the beads. So this is what we call a mass transfer limitation. So there are different kinetics that to predict, or different models that to predict how this substrate limitation is affects the enzyme activity. A third one, which is similar to the previous problem, is being unable to control the microenvironmental condition. In any model or any enzymatic reaction, we always predict, we can easily predict how we can perform. So there are so many models like a Michaelis-Menten equation, so many other models are available. But every model depends on the substrate concentration. You should know what is the residual substrate concentration in order to use those models. Here, as per this, you are unable to know what is the substrate concentration. Each point in the bead is having different substrate concentration. It create a heterogeneous condition in the inside the beads. So this heterogeneous condition is definitely affects your prediction and productivity. So obviously you need to account everything in order to develop a new model with respect to the enzyme immobilization. So these are all the problem. It's not in your hand. You don't know what is the temperature gradient happening inside the beads, substrate gradient. You don't know whether all the enzymes inside beads can be used. In some cases, the substrate is only penetrate till a point inside the beads. After that, it's unable to go. So there is an enzyme which is around the center of the beads is not at all utilized for the reaction. So there are so many factors that is you cannot control whenever you do the immobilization. So this is the problem that we need to account in order to build a model that predict your enzymatic activity and it also predict how much product is produced at the particular stipulated time period. So these are all the problem of the enzyme immobilization. In spite of all this problem, the immobilization methods is widely used in enzymes. As I mentioned in the first slide, so always there is a needed condition where this is give better results. As a overall, if you compare the activity of an enzyme in a free form and in immobilized form, in 95 to 98% of the time, only the free form has a better activity compared to the immobilized form. So, because of this, always the immobilized enzyme methods are used only at certain necessary conditions. So, it is not universal. So, with this note, I come to conclude this particular presentation. I like to say thank you.